Assalamualaikum. This video will explain about the synapse transmission. What is a synapse? Synapse is the junction where a neuron communicates with another cell across a narrow gap via a neurotransmitter for an electrical coupling.
This is a structure of synapse. So the first one we look into is the synaptic node, which is contain numerous of mitochondria that supply energy for the activities of neurons. Next, it also contains membrane-bound synaptic vesicle, which contain the neurotransmitter. The second one is the presynaptic neuron. As you guys can see, the nerve cell that carries a nerve impulse toward a synapse. So the third one is the synaptic cleft, which is, as you guys can see here, there's a gap between the postsynaptic cell and the presynaptic cell, which is 20 nanometer wide. So the fourth one is the postsynaptic neuron, which is in the nerve cell that carry a nerve impulse away from the synapse. It contains this, as you guys can see, the ligand-gated ion channel on postsynaptic membrane, has a specific receptor for neuron transmitter, and also allow movement of ion into postsynaptic neuron.
Okay, to continue, for neuron transmitter removal, it has two methods, which is reuptake of neurotransmitter. Neurotransmitter basically transported back into synaptic knob by active transport. And for the second method, it is enzymatic degradation by specific enzymes in the synaptic cleft. For an example, acetylcholine degrades acetylcholine, and monoamine oxidase degrades norepinephrine.
5. The binding of neurotransmitter to the receptor causes the ligand-gated ion channels to open on postsynaptic membrane. Third, the diffusion of Ca2+ triggers synaptic vesicle to fuse with presynaptic membrane and release the neurotransmitter into synaptic cleft via exocytosis. After the action potential at postsynaptic membrane is produced, the neurotransmitter is removed. It has two methods of removal. 4. Neurotransmitter diffuse across synaptic cleft and bind to specific receptors of the ligand-gated ion channels on postsynaptic membrane. Second is enzymatic degradation by specific enzyme in the synaptic cleft. For an example, acetylcholinesterase degrades acetylcholine, and monoamine oxidase degrades norepinephrine. First is reuptake of neurotransmitter. Many neurotransmitters are actively transported back into the synaptic knob that release them. Second, depolarization triggers the opening of voltage-gated Ca2+ channels. Presynaptic neuron membrane become more permeable to Ca2+. Ca2+ from extracellular fluid diffuse into synaptic node. First, action potential arrive at synaptic knob of presynaptic neuron, so presynaptic membrane undergo depolarization.